Hello and welcome to your own classroom. Today in the fast track revision series we would be learning about cell junctions. But before that, let's have a look at the basement membrane. What is a basement membrane? Basically, almost all the epithelial cells lie on the non-cellular basement membrane. Nutrients enter epithelial tissues from the underlying connective tissues by diffusion through the basement membrane. So let's see what I am talking about. As you can see in this diagram, there are two epithelial cells here. Let me mark them. So this is the first one and this is the second one. So there are two epithelial cells here, and these two epithelial cells, they are lying on a basement membrane. The green structure just beneath the epithelial cells, this structure is of basal lamina. This is one part of the basement membrane, and the black or thick structure lying beneath the basal lamina is reticular lamina. Basal lamina has got further two parts, lamina lucida and lamina densa. So basically the basal lamina is synthesized by or secreted by the epithelial cells, and the reticular lamina are secreted by the underlying blood capillaries. So these are blood capillaries. So these blood capillaries secrete the contents of reticular lamina. Now let's look at the component of basal membrane in detail. Basal lamina is the outer thin layer close to the epithelial cells. It is composed of mucopolysaccharides, glycoproteins secreted by the epithelial cells, and this layer is seen with electron microscope. And as we have seen that basal lamina is composed of two parts, lamina lucida. So it is a glass-like structure composed of proteoglycans, glycoproteins and integrins. Integrins are a kind of adhesion proteins which helps in the sticking of two cells together. And then there is lamina densa. Lamina densa is composed of mucopolysaccharide, proteoglycan and little amount of collagen. So basal lamina has got two parts. Now we'll talk about reticular lamina or fibrous lamina. Reticular or fibrous lamina is that in thick part that is formed by the secretions of underlying blood capillaries, and it contains collagen and reticular fibers in it, and it is seen with the light microscope. Since it is a thick layer, so it can even be seen with a light microscope. So what happens, as you can see in this diagram, the two epithelial cells, these are receiving nutrients from the blood capillaries via diffusion through the basement membrane. So nutrients are diffusing through the basement membrane into these epithelial cells. And why there is a requirement of basement membrane? Because epithelial cells are not directly supplied with the blood vessels. Now let's talk about the cell junctions. So we will look at some specialized junctions. The first one is intracellular bridges. Basically, intracellular bridges are the minute projections arising from adjacent cell membranes making contact with one another. So this is the first cell and this is the another cell. Both the cells are in contact, and how they are in contact? The first cell is sending some projections, and these projections are in contact with the projections of the second cell, forming a bridge-like structure, and this is referred as intercellular bridges. Next is interdigitation. These are basically finger-like processes of adjacent cell membranes fitting into one another. So this is the first cell, this is the second cell, and the processes from the first cell, they are kind of fitting into the processes of the second cell. So this is interdigitation, and it increases the area of contact between adjacent cells, helping the cells to adhere. These kind of junctions helps in the stimulus. A transfer of stimulus means if one cell gets the stimulus, it helps in the transfer of stimulus to the another cell. Now we will look at another kind of specialized junction, and this is tied junction, or it is also referred as blocking junctions or occluding. The meaning of occluding is same as blocking. So occluding junction, or it is also referred as Zonula Occludens. Remember it, it is called as Zonula Occludens. So what happens? Plasma membrane in the apical portion of the cells become fused to form tight junctions. The fusion is secured by sealing strand. Suppose these are two epithelial cells and the plasma membrane in the apical portion of these two cells, it somehow fuses, and this fusion is secured by proteins called as sealing strand. These barriers form a diffusion barriers, means tight junctions between two cells do not allow any fluid to leak between the cells. It is present in the intestinal brush border epithelium, endothelium of the brain to prevent the leakage of fluid between the adjacent cells. So let's have a look at this diagram. So as you can see, the two membranes have fused at a site here, and this fusion is like secured by sealing strand. It is a kind of protein which sits on the area of fusion so that it cannot open, preventing any leak of preventing the leak of any substance between these two cells. Now let's look at another kind of junction. This is called as adhering or fixing junction. So from the name itself, it's very clear that the junction which allows two cells to stick to one another. It is of three types. The first one is called as desmosome. It is also referred as macula adherens. These names are very important. Macula adherens, and it serves anchoring functions, means holding the cells in place. It is present in skin, cardiac muscle fibers, vagina and urinary bladder. So if you will look this structure closely, you can find two protein plaques. These black structures are the protein plaque present on the cytoplasmic side of the two cells which are in contact. From these protein plaques arises microfilaments of keratin, and these are referred as tonofibrils. These microfilaments and protein plaques and the other structures, basically they are helping these two cells to remain close to each other. And there are some adhesion proteins which are attached to the protein plaques on the outer side of the cells, and these between these two adhesion molecules, some extracellular matrix get trapped, which is shown in the green color here, this one. Now we'll talk about hemidesmosomes. Hemidesmosomes are also referred as single-sided desmosomes, and these are present in almost all the epithelial cells, and it helps in the joining of epithelial cells to the basal lamina of the basement membrane. That's why I taught you about the basement membrane in the beginning. So as you can see here, this is one epithelial cell here, and it is sitting on a basement membrane. Now the cell is connected to the basement membrane with a half desmosome like structure. So as you can see, this is a protein plaque, and from the protein plaque keratin microfilaments are projecting out, and these are the plaque is attached with adhesion proteins called as integrins, which helps in the attachment of these cells with the basement membrane. Now let's look at the third kind of adhering or fixing junction. So it is intermediate filament, or it is also referred as belt desmosome or Zonula adherens. Microfilaments of actin, it would be actin, ACTIN. Microfilaments of actin extend into the cytoplasm from dense protein plaques. So as you can see in this structure, the protein plaques are same as that of desmosomes, but the microfilaments that extend out of it is of actin. No intercellular filaments between the cell membranes, means there are no intercellular filaments between the cell membrane, rather adhesive material called as cadherin is present in between them. And this intermediate filament also serve the function of anchoring, and it is the location of these intermediate filaments is epithelial and endothelial cells. Now we will look at the last and the most important junction, and this is gap junction or communicating junction. Communicating junction helps in the exchange of chemicals and ions between adjacent cells, and these junctions connect the cytoplasm of the cells. Basically how they are formed, let's see. As you can see, there is a cylindrical protein called as connexin. 6 connexin proteins come together to form this kind of structure with a hole in between, and this is referred as connexon. And when you will see individual cells, so these cells, what they do, cells recruit connexons on their cytoplasm or on their membranes. So there are two cells, in these two cells are in contact with one another, and they have recruited connexons on their membranes so that cytoplasm or contents of the cytoplasm can be exchanged between them. Gap junctions are present in the intercalated disc of cardiac muscles. Intercalated disc of cardiac muscle fiber. So if you remember, cardiac muscles, two cardiac muscles, they are connected with each other by intercalated disc, so that the stimulus or the action potential between them transfers very quickly. And these intercalated discs have gap junctions, so that ions or specifically calcium ion can be transferred from one cardiac muscle fiber to another cardiac muscle fiber. It is also seen in case of smooth muscle fiber, single unit smooth muscle. Now, if you have gone through like the reference books of biology, you might have seen this diagram. So this is a diagram which is showing all the cell junctions at one place. In the top, you can see these are the microvilli present on the cell membrane, and then at the apical portion of the cell there is tight junction or zonula occludens. Then it is showing gap junction, and then zonula adherens, the location of zonula adherens or the intermediate filaments. Then there is macula adherens, interdigitations, and finally it is showing the basement membrane. So this that's all for this lecture. Thank you.